Alright, in this video we will resume our survey of biopolymers. We'll talk about proteins first, which are made up of amino acids. Here we can see the structure of a basic structure of amino acid. Amino acid because it has an amino group on one end and a carboxylic acid on the other, amino acid. In between those two groups there's an alpha carbon, and connected to that is a side chain, often talked about just as an R group. Now every amino acid is going to have this same backbone, and every amino acid is going to have a different side chain, so that's what's going to give them their different properties. Now in aqueous solution, amino acids exist as zwitterions, meaning doubly charged molecules with a positively charged amino group and a negatively charged carboxylic group. And in the next couple of videos we'll be talking about some of the consequences for its zwitterionic form. This is again two other depictions of the same molecule, ball and chain, and a space-filling model with the amino group, the alpha carbon, and the carboxylic acid. Now, proteins are linear chains of amino acids, and so that we're going to have to connect amino acid 1 to amino acid 2. We're going to do that by a type of reaction called a condensation reaction, where we'll lose a molecule of water, but we're going to connect the carboxylic group to an amino group of the second protein. So here is amino acid 1, here is amino acid 2. They're joined between those groups in this gray box by a bond called a peptide bond. This peptide bond, there's nothing special about peptide bonds compared to other bonds, but it's a convenient way to refer to that. Now, peptide bonds do have some properties that some of these other bonds don't. Even though this is a single bond, it has partial double bond character. So these four atoms will be planar. We will not rotate around these bonds, whereas these single bonds we can rotate freely. Down here we see three amino acids connected to it. This is the amino terminus because we have the free amino end. This is the carboxy terminus. We have a histidine, a cysteine, and a valine amino acid. Now you won't have to memorize the amino acids. As you can see, I kind of struggled a bit on that, but we will need to know a little bit about amino acids, and we'll get to that in a second. An important point about the synthesis of proteins, we'll see this a couple times, is that proteins are synthesized from the amino terminus to the carboxy terminus, sometimes depicted as N to C. Here we have the amino group. This amino acid was before serine, glutamine, and lysine. If we're going to add another amino acid, we're going to connect it to the C terminus, so that the amino terminus is always the first part of a peptide that is synthesized. Now I'm going to run through different types of amino acids. We're talking about, I'll just flip through charged amino acids, uncharged polar amino acids, and nonpolar or hydrophobic. So we're going to have to remember these three groups. Here are the charged amino acids: lysine, arginine, and histidine. These are all positively charged on their side chain. Histidine can be neutral or positively charged. Acidic side chains: aspartic acid and glutamic acid. These are all charged, so these will tend to have certain properties that you will need to be able to deduce and reason about when talking about that. For instance, just as a preview, you would never find a charged amino acid buried inside the hydrophobic interior of a lipid membrane, but we'll get to that. The second main category of amino acids are polar, like the charged amino acids, but they themselves are not charged. We have asparagine and glutamine that have carboxamide side groups, and we have three types of amino acids that have hydroxyl groups: serine, threonine, and tyrosine. These also tend to be exposed towards the aqueous solvent. These will not be buried inside the hydrophobic interior of proteins. These three amino acids with their hydroxyl groups can all be phosphorylated, and that will play a major role in cell signaling. We'll see those. The third type of amino acid are the hydrophobic amino acids. There are 10 of them. They are primarily hydrocarbons. As you can see here, methyl groups, hydrocarbons. We have a methionine with a sulfur group, and the largest amino acid, tryptophan, with an amino functionality. There are two special types of amino acids here. Proline, this is technically not an amino acid, you don't need to know this, but it's called an amino acid, and this has a side chain that is connected to both the alpha carbon and the amino group, so we're not going to be able to rotate around this bond, so that will have various structural consequences whenever a proline is incorporated into a protein. And then we have the smallest amino acid here, glycine, where the side chain is merely a hydrogen atom, and so that this is extremely flexible. Both glycines and prolines can introduce kinks into the folded shape of proteins. So again, if on a test you were asked, is leucine a hydrophobic or hydrophilic protein, or what class is it, you would have to know that it's a hydrophobic amino acid. Okay, that concludes our brief introduction to proteins. We'll now talk a little bit about nucleic acids. All right, nucleotides are the subunits of DNA and RNA. There are three major parts to it. There is a backbone, which is the phosphate group and the sugar group. Note, we'll talk about this a lot, the phosphate group is negatively charged. That means the backbones of DNA is going to have a lot of negative charges. And then the base, which is sometimes called the nitrogenous base, they are varying. We'll see that this is what varies, A, C, T, G, or U in the case of RNA. Now, there are two types of nitrogenous bases, pyrimidines and purines. In DNA, we have adenine, guanine, thymine, and cytosine. And in RNA, instead of thymine, we'll have uracil. But uracil and thymine code for the same information content. Each time you have a base pair in nucleic acid, you'll have one pyrimidine and one purine. The way I remember this is the longer word pyrimidine is the smaller structure, and the smaller word purine is the larger two-ring heterocyclic structure. The second part of nucleic acids is the phosphate group. In the final structure of DNA and RNA, we have a single phosphate unit, but the original building blocks, the nucleotides, are triphosphates, so they have 4 negative charges. We will lose 2 of those 3 phosphate groups, pyrophosphate, as the molecule is being synthesized. The third constituent of nucleic acid is the sugar, pentose. It's either ribose or deoxyribose. It's a 5-carbon sugar. 1, 2, 3, 4, 5. Used in RNA, we have this hydroxyl group at the second position, the 2' position, and in deoxyribonucleic acid in DNA, we've lost the oxy of the hydroxyl group, replace it with a hydrogen. So this is a small molecular change, but it has large consequences for function and structure of these molecules. Kind of amazing. Now, you might wonder why these are called primes, 1, 2, 3, 3', 4', and 5'. That's only to indicate positions on the sugar. The positions on the nitrogenous base would be without the primes. So we're going to talk a lot about the 5' carbon and the 3. Now there's a reason we always talk so much about the 5' carbons and the 3' carbon, 5', 3'. It's because it's between the 3' carbon and the 5' carbon that we have a bond via the phosphate group. This phosphate group will attack at this hydroxyl group, and we'll form a phosphodiester linkage between the bases, again, at the 3' carbon and the 5' carbon. You always add new nucleotides of the growing nucleic acid chain to the 3' end of the nucleic acid. Although we'll focus in the first part of the course, nucleotides' role in DNA and RNA, information storage and information utilization. Many of you will remember that these molecules can perform other roles inside the cell. In particular, you'll remember ATP is one of the main high energy intermediates that can be used for energy storage or utilization inside the cell. The ATP has, again, these 4 negative charges in close proximity. You know that negative charges repel each other, so for them to be in this close configuration, that's a high energy form that when ATP is cleaved from ADP into ADP, in this phosphate group will leave, you'll be relieving some of that negative electrostatic repulsion. And using that, hopefully capturing that energy to do some useful work. ATP can be part of a molecule that's a coenzyme used in enzymatic reaction. There are also forms where the single phosphate group will bridge between the 5' and the 3' carbon. This is called cyclic AMP, and this is a molecule that's a signaling intermediate. The last point to make about DNA in this particular lecture is a point you'll see over and over again. This is already the second time you're seeing it in this lecture, is that DNA is always synthesized from the 5' end to the 3' end. We're always going to add a new nucleotide down here. Here you see 4 nucleotides, G, A, T, and C, the 4 different bases you can have, and their particular orders how information is stored in the nucleic acid backbone. All right, that concludes our introduction to 4 biopolymers. Please be sure to answer the questions on the website prior to attending class.